In our previous lecture, we just began talking about the chapter on what we call biochemistry, which is the chemistry dealing with the compounds involved in biological processes, living organisms, both plant, animal, human. And we had just taken a look at the first group of the four major groups of biochemical species that we're going to look at. We mentioned we're going to look at carbohydrates, we're going to look at the lipids, we're going to look at proteins, and we're going to look at nucleic acids and their general structure. The first one that we mentioned and began to look at were the carbohydrates. And once again, the carbohydrates are those compounds that contain carbon, hydrogen, and oxygen. And specifically, as we saw yesterday, that these often contain the functional groups of aldehydes and ketones and alcohols. Those are the three functional groups that we see contained in the carbohydrates. The simplest of the sugars, the building block, the single unit sugars as we refer to them, are referred to as the monosaccharides. Of course, mono as a prefix means one, and the saccharide is the sugar unit. You might recognize the last part of that, the saccharide. One of the first artificial sweeteners produced was called saccharin, and the name saccharin came because it was a replacement for natural occurring sugar units. We started also by talking about the pentose sugars, which the pent as a prefix means, of course, 5-pentane. We've talked about earlier in organic chemistry. So the pentose sugars are those sugars containing 5 carbons. Now again, please let me mention, don't worry about drawing out all these structures as I put them up today. That's not the key thing. The key thing here is that we have a 5-carbon sugar. Okay, so if you recognize pent, you would recognize the 5 carbons. This particular sugar does have, hopefully you could recognize that group up there, that's the aldehyde group. And of course the OHs over here are the alcohol groups that we've talked about previously in organic chemistry. Now, even though we can draw the molecule in a linear form, and even though it does exist occasionally in the linear form, by that I mean that carbons are all hooked up in a line, more frequently in nature we find the pentose sugars, and actually we'll find that we find most of the sugars in what we call a heterocyclic form rather than in a linear form. Now, that's not a new term to us because we did talk about heterocyclic when we talked about amines. The term heterocyclic, of course cyclic you could guess, that means that we're going to hook things around in a cycle. But the term heterocyclic means that one of the atoms in the ring is a non-carbon. Okay, homocyclic would be something like the benzene that we talked about in a previous lecture. The benzene, the 6-carbon ring structure, is a homocyclic structure. Heterocyclic means that one atom in the ring is a non-carbon. So if we had something like we had looked at earlier in the year, we looked at something like this with a hydrogen on here and a pair of electrons, and we called that a heterocyclic amine. That was an amine, the nitrogen-hydrogen structure in the overall. This is heterocyclic, and as I say, in most cases we really find the pentose sugars then not in a linear form in nature, but rather in the heterocyclic form. So let's take a look at the pentose sugar then in its heterocyclic form. We have two types of pentose sugars, and we're going to come back and talk about these even further when we talk about the nucleic acids. This is called the ribose sugar. And actually, the ribose sugar is exactly what we had just a moment ago in the linear form, except that we have bent it around and hooked it together in that oxygen that was there on the aldehyde group. And then we still have an alcohol, an alcohol, an alcohol, and an alcohol. We had the 4 alcohols like we did previously. This particular form is called ribose. That little B in front of it is a beta ribose, but that's not that essential to us. So we'll only concentrate on the ribose part. The ribose, and then the next one to it, which is also a pentose sugar. It still means it has to have 5 carbons. And the carbons in this case, there's 1 carbon there. Each of these corners here are actually carbons. So that's where our 5 carbons are in that structure. And over here, of course, we would have the same thing. We have these 4 corners are carbons, and of course then the 5th one up there. So they're both pentose sugars. Ribose, and this is called deoxyribose, and the name deoxyribose comes from the fact that if you look right here, you notice that there's 1 less oxygen on this molecule than there is on this molecule. There's 1 less in alcohol group, essentially, in the 2. And so the name deoxy, when we talk about detoxification, meaning to remove something that's toxic, deoxy means minus an oxygen, and so that's where the name came from. It's the ribose sugar minus the 1 oxygen, and that makes it deoxyribose sugar. Now, these two, as I say, are important when we talk about nucleic acids. There are 2 nucleic acids, DNA and RNA. And RNA stands for ribonucleic acid. And the R comes from this part right here, ribonucleic acid. DNA comes from deoxyribose. And so that's where the name DNA comes from, deoxyribose nucleic acid. Now, of course, this is just one component of each of these nucleic acids, and we'll look at the nucleic acids in greater detail later. But our starting block then, our simplest of our sugars, are then the pentose sugars. Now we'll go up 1 step, and we'll go to the hexose sugars, meaning that we're now looking at something that has 6 carbons. We have 6 carbons, 1, 2, 3, 4, 5, 6 carbons. Notice that every one of the carbons has an alcohol group on it, and the end group up here is once again an aldehyde. As a matter of fact, this particular sugar is referred to as an aldohexose. So the aldo tells us that it has an aldehyde group on it, and the hex, of course, says that there's 6 carbons there. So the name tells us what the structure is. The common name for this aldohexose is glucose, or sometimes it's also called dextrose. Both of those, the dextrose is the just very generic common name, but glucose is the more common name. Sometimes this is also called grape sugar. This is the sugar that we find in grapes. This is the sugar that is going to be fermented to ethyl alcohol when we produce wine. This is the material. Remember when we looked at the beer and wine making. In an earlier chapter, we talked about the fact that in beer making, we are starting with like starches and must convert them to sugars and then ferment them. When we're making wine, we're already starting with a natural sugar that's there in the grape juice, and all we're doing then is bringing about the fermentation process. So, aldohexose, an aldehyde, 6 carbons, and the grape sugar. We also have a ketohexose. Notice here, again, we still have the 6 carbons, and we still have 5 alcohol groups, but notice this time this carbon double bond oxygen, instead of being on an N carbon, which is typical of the aldehyde, is on a carbon in the ring, in the chain. And so this makes this then, instead of being an aldehyde this time, this is a ketone. An R group, an R group, and the C double bond O for the ketone. So this is referred to as a ketohexose. And again, the keto tells us we're looking for a ketone in the structure, the carbon double bond oxygen in the ring, or in the chain, and hex, that we have the 6 carbons. This is called fructose, also known as levulose, but fructose, and it's fruit sugar. It's the sugar that we find in the rest of the fruits. So we have the grape sugar, if we can get both of them on here fairly close to each other. We have the one that occurs in grapes, which is the aldehyde, and the one that occurs in most of the other fruits. Apples, pineapple, peaches, pears, watermelon is a fructose, fruit sugar, and it's a ketone structure. That's the only difference in the two. Okay? All right. Any questions? Now, looking at the structure that we typically find them. Again, the linear is nice looking at to see the functional groups. But in reality, most of these still occur in nature in a heterocyclic form. So we're going to show these now in the heterocyclic form. The one here in the middle is the fructose. Now that means that it's still a 6 carbon. We haven't lost any carbons here. And we see here's a carbon, and there's a carbon, and then there's 1, 2, 3, 4 more carbons right there then in the ring. But notice this ring in the fructose now has 5. It's a 5-member ring, whereas in the 2 glucose molecules we have a 6-member ring. Carbon, 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 and the oxygen. And of course the same thing here. The 5 carbons in the ring and 1 carbon outside of the ring. In the fructose we have 4 carbons in the ring and 2 carbons outside of the ring. Now, there is, because whenever we make a ring, whenever we have a double bond, we talked about the fact that we have a restricted geometry. And when that happens, we can have geometric isomers. And so if things point up or down off of a fixed ring structure, that ring can't twist. So if it's fixed like a pie plate, whether we have something sticking up from the pie plate or down from the pie plate makes a difference. It's a different structure. And so here we see now on the glucose, okay, we had the 2 hexoses. This is the ketohexose, fructose. This is the aldohexose, which is the glucose. But notice we're talking about an alpha form and a beta form. Now the difference between the alpha and the beta form, we can see here, is that an alpha form, the OH on the number 1 carbon, that's this one right here, is below the ring. Notice that the OH is pointing down. Over here on the beta glucose, the OH is pointing up. And you might say, well, so what? I mean, big deal. So one is down and one is up, right? What possible difference could that make to anybody? Well, we'll see in a minute that it makes quite a difference in the additional bigger sugars that we make, whether we make them from an alpha glucose species or a beta glucose species, we get totally different materials produced. And so, yes, in fact, it becomes quite important to us and to understanding the more complex carbohydrates. Now, glucose is sometimes referred to as a reducing sugar, and that doesn't mean that we lose weight by eating it. What it means is that it has the ability to cause something to be reduced. We talked about oxidizing and reducing agents in chapter 13. A reducing agent was something that caused something to be reduced and itself be oxidized. Okay? So a reducing sugar is something that will cause something in the biological system to be reduced. For instance, it might reduce iron 3 to iron 2, or it might reduce various other metallic ions that are part of our mineral needs. It might reduce them from, say, a copper 2 to a copper 1, things like that. So it's a reducing sugar. When it causes something to be reduced, it itself must be oxidized, and we see what happens up here is this aldehyde group becomes oxidized. Notice that we've added an oxygen on here to now the organic acid. That's a carboxylic acid group. So it has been changed then from an aldehyde to an organic acid by causing something else to be reduced in the biological system. Glucose also can undergo oxidation. And here's the case where it undergoes oxidation. So here we see now that we again start with the aldehyde on the glucose, and this time we're going to add a hydrogen here. So we're breaking that bond and putting a hydrogen on and converting it into an alcohol. Well, this particular substance here with 6 alcohol groups, no aldehyde or ketone, has a commercial name called sorbitol. This is a fairly good sugar substitute. It has about 70% the sweetness of table sugar. But one of the advantages is that when the biological system processes it, that we don't end up forming the normal sugars that we do. Most of it is converted to carbon dioxide and water, and it can be used by persons that have a concern about diabetes. It's not a non-caloric, it's just a sugar substitute. Biologically, it goes through a different process. One, it undergoes then the change to CO2 and water, which allows it to be used as a sweetener by people that have a problem with diabetes. So again, the sucrose being converted to this is used as a sweetener in candies. It's used as a sweetener in gum material. So it's a fairly common sweetener overall. Now, that's the simplest sugars. Now we're going to go to the next bigger group, and we're going to go then to now what we call the disaccharides. Well, we all know what the prefix di means. Of course, it means that there are 2. So we're going to take 2 of those monosaccharide groups that we just talked about, like a glucose and a fructose, or a glucose and a glucose, whatever the case might be, and we're going to hook 2 of them together, and we're going to make a disaccharide. Actually, I should have mentioned that on the monosaccharides, of course, we do have the glucose, we have the fructose, and then one that I didn't mention was, I'll just put it up here in the corner, one other monosaccharide is called galactose. And we'll run into that here again, but that's the third monosaccharide that we typically talk about. Glucose, fructose, galactose. Okay? Now, if we take a glucose and a fructose, two monosaccharides, two separate molecules, and if we reach in here and pull out a molecule of water, we dehydrate the sugar. And if we do that, what we end up with now is this structure right here. Notice this is the bridging oxygen. When we pull these off, it's going to be hooked from here to that oxygen. And this is sucrose. So the combination of a glucose with a fructose gives us sucrose. And this is our normal table sugar. Now, we'll talk a little bit about the digestion of carbohydrates in just a little bit, but our body can only absorb monosaccharides. As far as putting then energy into the bloodstream that can be taken to cells and used, it has to be in the form of a monosaccharide. So if we eat table sugar, before we can use that table sugar, it has to be rehydrated, the process has to be reversed, and the sucrose has to be converted back to the fructose and glucose. Once it's converted back to that, then it can be absorbed into the bloodstream and transported and used for energy. Now, honey, natural occurring sweetener, but honey is about a 50-50 mixture of glucose and fructose. Now, bees have an enzyme in their system that allows them to take sucrose. They go out to the flowers and pick up the liquid from the flowers, which contains the sugar, and part of the sugar that they don't use, the excess sugar, is automatically broken down into these two and stored as honey, which is then this mixture here. So if you want quick energy, then eating a candy bar is going to be a slow process because you're going to have to digest this first. But if you want quicker energy, if you eat honey or honey-containing sweetened products, they're already ready to be absorbed into the bloodstream. So the digestive process doesn't have to occur. All right? Glucose and fructose will give us sucrose or table sugar. Another combination is a glucose plus a glucose. An alpha glucose plus an alpha glucose gives us a sugar, a disaccharide called maltose. Now, maltose is a sugar that we talked about earlier in the course when we again talked about the beer brewing industry. Malt, which is a part of the brewing process. Malt contains maltose. Maltose is the first sugar that we're using in the beer brewing industry, which will finally be broken back down into glucose. And then once it's in the form of glucose, it will begin undergoing the fermentation process to ethyl alcohol. So this is malt sugar, or sometimes referred to as grain sugar, because it's obtained when we germinate the barley, and then we take the germinated seed and dry it. We will have this malt sugar. So this is sometimes referred to as grain sugar. Okay, and the grain sugar then is, as I say, what is used to start the brewing process, or also is used as a sweetener in malted milks. Okay? So when we talk about a malted milk, we're taking the milk and the ice cream and the flavoring, and then we add this, which makes it slightly sweeter due to the presence of the malt. Another combination, and this one I don't have drawn out, but if we take a glucose and a galactose, we produce another disaccharide called lactose. And lactose is known as milk sugar. Okay, this is the sugar that we do find in milk, which of course is one of the major energy ingredients for babies, for youngsters, for young animals. A very high level of lactose, which is used by the body then to provide the sugar content. So we had 3 monosaccharides. We had the glucose, fructose, and galactose. And if we hook different combinations of these together, we can produce various types of disaccharides. 3 that we've listed here then. The first one, sucrose or table sugar, maltose or grain sugar, and lactose, milk sugar. In the text is a table that shows the combination and the different types of disaccharides that one gets. Now, when we think of sugars, we think of course, sweetness, and this gives a table of the values of the contrast in sweetness of the various carbohydrates. Here is sucrose, table sugar. It is arbitrarily set as a sweetness of 1. In other words, there's no way to measure this directly other than by taste. And so the sweetness is in terms of palatability test. And so sugar, table sugar, is considered 1. We see that fructose or fruit sugar, this is a monosaccharide. So let's identify the ones here that are the sucrose is a disaccharide, fructose is monosaccharide, glucose mono, galactose mono, and then di and disaccharides. All right, so we see that the sweetest sugar that we have, the sweetest carbohydrate that we have, is fruit sugar or fructose. Glucose, 0.74. Galactose, about the same as maltose. So the malt sugar and galactose are about the same. And finally, the milk sugar is not very sweet at all. So you don't really detect any sweetness when you drink milk, even though there is that carbohydrate in there that would have some sweetness to it. It's a pretty low sweetness when we compare it to table sugar. All right? Now the last 3 are artificial sweeteners. Cyclamate, about 3 times sweeter than table sugar, and then NutraSweet or aspartame, and saccharin. Saccharin was the first of the artificial sweeteners that were produced. And as I mentioned, the name saccharin, of course, comes from the term saccharide, which was the term used for sugars. Saccharin, extremely sweet, and it was developed primarily in World War II when sugar supplies were short, and what sugar we did have went to the war efforts, and so people still wanted sweetener for their cereal and things like that, and so somebody set out and formed the saccharin. And notice the sweetness. Obviously, you can't put a teaspoon of saccharin on your cereal. As a matter of fact, it was sold originally as a liquid, a very, very dilute liquid, and you put it on as a drop. So if you had a bowl of cereal, you'd take a drop of saccharin and put into your bowl of cereal for the sweetness. The one problem with the saccharin was that it does leave sort of a bitter aftertaste. And so from that standpoint, it was not too popular. And so therefore, in the 50s, they set out to find a different sweetener, and this one happened to be cyclamate. And it's not as sweet. As a matter of fact, it's 100 times less sweet, but it's still 3 times sweeter than table sugar. So you'd have to use a fair amount, but you didn't have to cut way back like you did with the saccharin. Somebody did a study in Canada on rats and found that if you fed a rat 100 pounds of cyclamate a day, that it died from cancer. Well, you probably feed it 100 pounds a day of anything and it would die of something. But the study then led to some concern that cyclamates, which were being used extensively in dietary drinks and artificial sweeteners and low fat or low sugar candy, low calorie candy, things like that. So the federal government ruled that cyclamates could not be used for human consumption, and based on Canada study. There was a lot of debate about whether the study was valid and can we test with this magnitude of chemical to really determine toxicity. But they made a move that we couldn't use it anymore. But they really weren't as worried about us as we might have thought, because somebody had already discovered aspartame, which is 200 times as sweet as sugar, and doesn't have the bitter aftertaste of saccharin, and didn't have any apparent health problems. And so cyclamate was removed from the market, and of course most of the sweetening today we see is artificial sweetening we see today is done with NutraSweet. However, later study took away the villain part of the cyclamate, and so you will see now cyclamates once again being used as sweeteners. And because of the bitter aftertaste here, by using a very small amount of this with cyclamate, you can get then a fair sweetening with a relatively small amount and get rid of the bitter aftertaste primarily. And so a combination of the cyclamate, saccharin is one of the things that's used for sweeteners. The other major artificial sweetener is, of course, the NutraSweet. But again, the sweetest of all of the natural sugars is the fructose. Now, the next group of carbohydrates, if we started at mono and then we went to di, and then we could go to tri, but we're going to jump further than that. We're going to go to poly. Poly means many saccharides. Okay? So let's take a look at some polysaccharides. Whoops. Now, the polysaccharides mean that we have many of these single units hooked together, and they're all done by dehydrating, by pulling a water molecule out from between the monogroups as we go along. If we go back, let me do that, I guess, quickly with the disaccharide here. Notice that when we pull out a water here and hook it together here, notice we still have an OH group here and an OH group here, just like we had there in the monosaccharide. And so those OH groups are still available to attach to another one on this end and another one on that end. And so we can propagate this into a polymer, a large then molecule. This is where that little difference between the alpha glucose, remember the OH pointing up, and the beta glucose, the OH pointing down on the ring on that first carbon, turns out to be quite important. If we hook together alpha glucose strings, so this is a monosaccharide and there's one hookup and then the next one hooked up and the next one and the next one, etc. And this produces what we call amylose. And this is a type of what we refer to in general as starch, amylose. Another form of starch occurs when occasionally, instead of everything hooking together in a continuous straight line, occasionally we get one from a carbon 1 hooking to a carbon 6 structure. That's called the 1,6 linkage. Not that you need to worry about that. But when we do that, we make another starch called amylopectin. If you've ever heard about pectin from fruits, we use pectin to make jellies. We're adding this particular type of starch, which then in the cooking process, and when it cools, helps form a semi-solid mass, so our jelly sets up, so it's not like syrup, but it becomes like a semi-solid. This is the pectin that we talk about, the amylopectin here. Now, these are the two common starches, and they're made from, both are made from that alpha glucose. That's the important part here, the alpha glucose. Our body, if we eat a lot of sugar, a lot of disaccharides, for instance, and we don't need all of that for the energy, our body takes those disaccharides and begins hooking them together, and hooks them together in the form of a starch, amylopectin. In the body, we refer to this as glycogen. This is the term that is used for the stored starches or the stored carbohydrates in our body are referred to as glycogen. And glycogen is merely amylopectin, which is just our body putting together all those mono and disaccharides that we don't need and storing them someplace as then this starch, stored material, glycogen. And then, of course, when we become more active and the body demands more sugar, then the body rehydrates these, starts clipping off little pieces of it and puts the monosaccharides back into the bloodstream so that it can be used to provide us the energy we need. So we store the excess sugar in our bodies in the form of glycogen. Now, if in contrast, I'll put this up. This just gives another little picture of the two. This is the amylose. It just kind of makes up a chain like hooking paper clips together to make this big molecule. And then the amylopectin, which is a branched. So every once in a while we hook another set of paper clips going off in a different direction, and so we get this branched, and this is the amylopectin then, or glycogen, that our body stores the excess sugars. Now, if in contrast, once again remember, alpha had the OH up, beta has the OH down. If we take beta glucose and hook it together, instead of making a line with the oxygen down at that V like it did in the starches, notice that the oxygen now is hooked this way, upward, 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 and so we end up with a stair-step looking molecule, and this is cellulose. Both of these are made from monosaccharides, starch and cellulose. The only difference is the starches are made from the alpha glucose and the cellulose is made from the beta glucose. Now, again, of what importance is that to us? Well, the fact is, in our digestive system, we don't have any enzymes in our digestive system that allow us to break cellulose down. So if I eat foods containing cellulose, which is basically just another modified form of starch, a polysaccharide, if I eat cellulose, it does nothing. It goes right on through the body. It's flushed out. It's waste. We cannot use it for energy. Now, there are some organisms that can. For instance, termites which have associated with them a bacteria that does have an enzyme that can break this down. So the termite goes out and chews up the wood, and then the bacteria provides the enzyme to break it down, and so both the microorganism and also the termite can use then the monosaccharides produced in the process to live on. But most animals, humans, cannot process cellulose. We don't have the enzyme necessary for it. So there's quite a difference whether we take an alpha or a beta glucose and hook them together to make our polysaccharides. Of course, here's an example of a couple of places that we have a good supply of polysaccharides. Of course, the potato for the edible starch, and cabbage. Cabbage has both a fairly high amount of carbohydrate and also cellulose, by the way. But it turns out that we do like to have some cellulose in our diet because we call it roughage, and it helps the digestive tract, stimulates the digestive tract. And so it's not bad once in a while to eat a wood pencil just to kind of stimulate things a little bit. Okay. Of course, the major place that cellulose is found in nature, of course, is in plants, where it is the structural material. It's the material that gives the tree its support, the leaf its structure. It's the cellulose. So it looks like basically starch, but because of its structure it is a very rigid and can provide then considerable support. I mean, the support that a tree branch can hold is relatively amazing. And it's just these fibers of cellulose that are then hydrogen bonded together that make up these giant strands that hold basically those or make the structure for those plants. All right. Now, a little bit about digestion quickly and what happens when we eat something. And let's start with a polysaccharide, because usually a good share of what we eat is starch. In the mouth, we have an enzyme which will begin the conversion of the polysaccharides now to maltose. In other words, we're going to take the big molecule and clip it into a disaccharide. Maltose is a disaccharide. Now, you can kind of demonstrate this yourself if you take an unsalted cracker or unsalted bread and put it in your mouth and just chew it, let it slush around for a while, and then taste it. And you'll notice a sweetness. The sweetness is due to the fact that the enzyme in the mouth has begun the conversion to the malt sugar. And so what you're detecting then is maltose. Now keep in mind it's only about 30% as sweet as table sugar, so you're not going to get a real large abundance of sweetness, but you'll notice a definite sweet taste if in fact you have an unsalted source of starch in your mouth. As far as the carbohydrates are concerned, the stomach doesn't do anything. There's no digestion of carbohydrates in the stomach. So it has to get through the stomach before we really get to have any advantage of the starches and things like that that we eat. Once it moves into the small intestine, then we're going to find a whole series of things begin to occur. First of all, and we talked about enzymes in the chapter on things that affect reaction rates, but in the biological system, remember that each one of these catalysts, each one of these biochemical catalysts is referred to as an enzyme, and each enzyme has a name, and it ends in ASE, and it usually refers to what it's doing, okay? So a ferroxidase was one I think we looked at one day. That was something that catalyzed the formation of the iron II in the heme. Okay, so the name. Now, here we see then, we start, starch reaches the small intestine. We have an amylase. Okay, that means an enzyme that is going to break up, going to chop up amylose. Remember, amylose is the name for the polysaccharide starch molecule, and it's going to break it up into units of maltose. As the maltose continues to move through the small intestine, another enzyme comes into play, and the maltose, in the presence of maltase, becomes cut in two and produces the two glucose molecules. Now, as soon as the glucose is formed, then it is now ready to be absorbed into the bloodstream through the walls of the intestine, which are very rich in blood vessels. So at this point, the glucose can move into the bloodstream, begin being circulated to the body. Now, as I mentioned, if I take some table sugar and eat it, it has to pass through the mouth, through the stomach, into the small intestine, and get to the point where it runs into this enzyme called sucrase before it's broken down into glucose and fructose, which can be absorbed into the bloodstream. If I eat fructose or honey, which is a mixture of these two, if I eat honey directly or eat an apple or a peach or a pear, something that will give me the fructose, then that is already ready to go into the bloodstream as soon as it passes from the stomach. Still has to get out of the stomach. I mean, so we don't get an instant. If we eat something, it's still got to get through the stomach before it can be absorbed into the bloodstream, and then we get our energy. But we don't need to wait for this process to occur, because it's already in this form if we eat honey. All right, the sucrose undergoes breakdown, and the lactose or milk sugar undergoes breakdown in the presence of this enzyme called lactase. Now, when we're born, we have a very high level of lactase. And of course, that's necessary to break down the milk sugar, because milk is a major part of our diet. As we mature, our body decreases the amount of lactase it produces. And in some cases, as adults, certain people will get to the point that they'll have no lactase, and therefore, if they eat milk products, pizza, ice cream, all of those good things, the lactose that's there from the milk products cannot be digested. And the lactose will pass into the large intestine as lactose molecules. There are bacteria in the large intestine, however, that will begin to ferment the lactose, thus causing tremendous amounts of gas being produced, stomach cramps, diarrhea, can be serious problems. And we refer to people that have that condition as being lactose intolerant. And of course, fortunately, and I think I mentioned this when we talked about the enzymes in the catalyst, fortunately, we've been able to synthesize this so that people with biological lactose intolerance can take a lactase tablet with their ice cream cone or with their pizza or with their cheese sandwich, and that will provide the enzyme to be able to break that lactose down into glucose and galactose so it can be taken into the bloodstream and not have the fermentation process. All right, in our next look in chapter 14, we'll look at the other three: lipids, proteins, and nucleic acids.